indulgence of the chair of the session, I asked to change my title, which is because I have the two graduate students who've done a lot of the work on modeling ischemia in my group are actually here, so I didn't want to steal their thunder. So I'm going to talk about some work we've done on modeling hypertrophic signaling pathways. These are the two people who are here, who won't thank me for putting up their pictures, but the work that they've been doing on putting together a cellular model for ischemia, modeling the different transporters and the energetic state of those transporters, is, I think, really beautiful work, and it's described in two posters, Kenneth Tran and Jonna Tuckelson. Please do go and see those posters for more discussion about our work on ischemia. But I want to talk about work that we've been doing in Auckland on modeling hypertrophic pathways. This is work, if I start with the acknowledgements lest I forget them at the end, work done by Mike Cooling, who was a PhD student and published in a couple of papers in Biophysical Journal. So hypertrophic signaling pathways, this is a project which is a modeling study looking at other things that calcium is doing in myocytes. Okay, so we know a lot about calcium in terms of coupling excitation and contraction, but calcium is also involved in many cell types, myocytes not excluded, as being a second messenger involved in regulating other processes, including regulating transcription. And so in hypertrophic pathways and hypertrophic responses, we have at least the possibility that calcium is involved in switching on gene expression. So first of all, I'll say just a little bit about hypertrophy. I think a lot of people will know a great deal about that. So hypertrophy, the heart does more work, then the heart gets bigger, not because cells are dividing, but because cells are gaining in mass. And this is a perfectly normal response to exercise in athletes and what have you. But also, it's there is a pathological side to the story. So in decompensated hypertrophy, the heart gets bigger, but not in a beneficial way. So this leads on to lots of risk factors for disease. You can stimulate hypertrophic growth of cells simply by putting on agonists such as endothelin and angiotensin. So this is data from not from us, but data showing that if you take a cell population and simply add, in this case, endothelin, then the cells get bigger. So there are a huge number of pathways involved in hypertrophy, pathways which have been identified in different cell types and have been thought to be involved in hypertrophic response. Just one of those pathways here involves calcium, but this is strongly implicated and is the one that we're studying. We want to ask several questions about this pathway, in particular in the context of the heart, not least because there are very different responses to this pathway which are found in response to different agonists, in particular endothelin-1 and angiotensin, for which there are data, but also because there's this overall question about how calcium can be doing different things in the heart. So calcium, we know, is providing this cytosolic calcium transient, which is coupling excitation to contraction, but it's unclear, and certainly there's a lot of discussion in the calcium signaling literature about how calcium can be doing these different things. How can calcium be, on the one hand, coupling excitation and contraction, and on the other hand, doing something like regulating gene expression? How can it perform these different roles? Is it a modulation of frequency? Is it modulation of amplitude? What's going on? And in fact, in this situation, people have put forward the idea that there really must be a different domain of the cell, a different specialized sub-location in the cell, where there is calcium involved in this pathway, which is somehow kept away from EC coupling. And so what we wanted to show in a modeling study is really that's not necessary. This has recently led to some interaction, or the beginnings, I hope, of some interaction with experimentalists actually just down the road here in Cambridge. But let me tell you about this study. So I want to ask those questions. First of all, how this pathway is regulated, in particular the response to different agonists, and secondly, how we can understand that calcium is performing these different roles. So here's a very schematic picture of our modeling. Our modeling here is mass action kinetics. We've parameterized it for endothelin. So this is modeling in much the same way that we've just heard about from Jeff. Modeling the receptor kinetics and then coupling this is a G protein couple receptor, coupling the receptor through the G alpha subunit to the production of the other intracellular signaling molecule, IP3, from PIP2 under the activity of phospholipase C, which gets activated downstream of this receptor. So we can model that process, and there's just about enough data to parameterize this, just about enough parameters in the literature to parameterize this for endothelin. So we're plotting the IP3 transient measured as a function of time, and we can fit that reasonably well. And we can look at things like dose-response curves and get kind of reasonable-looking dose-response curves, the right sort of thing anyway. So first question then is, how does this pathway respond to different stimuli? This is difficult because parameters are not really known for angiotensin. So what we've done is to do a sensitivity analysis to try to identify which components in this little part of the pathway might be responsible for the differences seen between endothelin and angiotensin responses. So sensitivity analysis means take the model and identify which parameters in the model determine things like the peak of the response, the width of the response, and so on. And by doing that, what we can find is that there are parameters in the kinetics of the receptor, which is really logically the only place in which we'd want to find these kinds of parameters, which can give you the differences between endothelin and angiotensin responses. So there's no other regulation of this pathway required to explain the differences for these two agonists, which perhaps isn't very surprising, and really it's only when you look at the dramatic differences in timescales of responses that you get for these two agonists that this really seems to be important at all. But it simply can be explained by differences in receptor kinetics. So what we've done then is we've built, in order to address that second question about how calcium can be doing these different things, we've built the rest of the pathway leading to IP3, leading to calcium release from stores, intracellular calcium stores of some sort through IP3 receptors, to activation of calcineurin by calmodulin, calcium-activated activation by calmodulin, and we can fit data there, and then look at the downstream effect. So this is now the transcription factor NFAT, which we know to be at least one of the transcription factors involved in mediating hypertrophic response, so producing proteins in response to these hypertrophic signals. And so what we can do with our model is we can look at two different pools. First of all, NFAT, which is in the cytosol, and then the second pool, NFAT, which is in the nucleus. NFAT gets translocated to the nucleus by activated calcineurin, or by forming a complex with activated calcineurin. And again, we can fit data to, well, kind of roughly follow what the experiments are doing. But the reason for doing all of this is to be able to look at this figure here. So now I will explain this figure. Along the bottom, we've got the strength of the calcium coming from this signal transduction pathway. So this is the amount of calcium that is coming from the response to the hypertrophic signaling. On the y-axis, we've got the percentage of NFAT which has been activated and moved into the nucleus. So this is, if you like, a surrogate, a measure of the amount of gene expression that's happening. And what we've got in these rainbow colors, so across from left to right, we are increasing the amount of hypertrophic signaling, if you like. And what we've got here in these rainbow colors are different strengths of EC coupling, so different calcium signals which we've got going on at the same time in the same compartment, different pacing with different frequencies. And so what you can see is that as we change, so not only can we see that we can activate gene expression using a calcium mediated signal in the same compartment as we've got EC coupling going on, it can happen even at very high pacing frequencies. So my conclusions are that, first of all, we can look at little changes in the receptor kinetics can explain differences in the calcium signaling in hypertrophy that we see for angiotensin and endothelin. Secondly, and perhaps obviously to some people in this audience, but certainly not obvious in the literature around this topic, is that molecular machinery, some of these proteins which are interacting with calcium sensing proteins, are doing reasonably interesting things. They're filtering out different aspects of calcium signals. So they're able to filter out, in this case, filter out the ECC oscillations and really only respond to the change in sort of calcium that you get from the hypertrophic signaling. Now, why is this important? It's important because people have identified not only can the ECC, the excitation contraction coupling calcium transient, be seen at the nucleus, calcium goes into the nucleus, of course, but this recent paper from just from earlier this year has now shown that this pathway, the IP3 pathway, is indeed the pathway that is required to get hypertrophic signaling as measured by hypertrophic proteins, and that you can get this hypertrophic response without ECC coupling. And what they've identified now is that these stores, the calcium release, the IP3-related calcium release, is from ER actually located near the nuclear envelope. So this is definitely a place where the calcium transient from EC coupling is definitely seen, and that this signal is happening at the same time with calcium released from the ER in the perinuclear region. And so what we've shown is that you don't need to come up with any other kind of explanation as to how calcium is doing these two things at once. Simply these proteins are able to, the calcineurin calmodulin system is able to interpret changes in calcium due to signaling over those oscillations. So I will leave you then with those acknowledgements and 2 posters for those who want to hear about our work on ischemia. Thank you.
Thank you.
Yeah, Edmund, I have a question. Do you get correct time kinetics of development of heart failure with this method? Because it usually takes a long time.
Yeah, no, I'd love to be able to address those sorts of questions. All we've looked at is this very early step in the pathway. So we are modeling at the moment is stopping when NFAT gets to the nucleus, and we haven't gone any further. But I'd love to be able to look at those sorts of questions. But yeah, I think at the moment there aren't data to do that, certainly in a kinetic, using kinetic modeling.
I think Guy Salama.
I was intrigued, of course, because what happens to calcium concentration in the perinuclear membrane? So your modeling is predicting an elevation, of course, which has the, what's the range and what is the relationship between calcium and the triggering signal?
Well, how much calcium?
Yes, what's the concentration, and is there a gradient between nuclear and cytosol?
So, well, it's probably best to answer that from the data from this paper that's just appeared rather than the modeling. What the modeling was trying to show is that you don't need to compartmentalize these 2 different processes into different parts of the cell and say somehow they're not talking to each other, they don't see each other, and so there is no problem to address, which is one of the things that had been, I guess, pushed in the literature. What seems to be happening in the perinuclear region, following the paper from Lou Roderick and colleagues at the Babraham Institute, is that the ER in the vicinity of the nucleus is the location of these IP3 receptors, and so what's happening is that the signaling, the hypertrophic signaling, is releasing calcium through IP3 activating those perinuclear receptors, and so you'd have a rise in calcium of sort of hundreds of nanomolar, so tens to hundreds of nanomolar of calcium. So certainly a large enough calcium rise to get the calmodulin calcineurin system to respond to it, and they've basically been able to show that is precisely the mechanism which is at play. And I think what we're showing is simply that these 2 things can live quite happily next to each other, because we know that if you measure calcium next to the nucleus, you see the calcium transient from EC coupling as well.
So 100 nanomolar is not much at all.
Yeah.
Based on cytosolic transients in a normal heart.
Yeah.
So why should that be any different when the perinuclear membrane is seeing those oscillations of calcium to even higher levels during normal contractions?
Right. So the point is that the calcium sensing proteins, which are activating transcription, are operating on timescales such that they are not responsive to the calcium oscillations. They're not responsive to the frequency of EC coupling, but they are able to respond to these slower but smaller changes, much slower transients in calcium from EC coupling, from ET coupling, from the hypertrophic signaling.
Andrew, I do apologize.